Ladies and gentlemen, welcome to this Jubilant Biosys webinar on the added dimension of scale-up. The promise of Biosys is captured under the concept of what we call 4D, the fourth dimension, time. We believe that time is life and should be treated with utmost importance. Especially in drug discovery and development, the importance of time has increased over time. And in this webinar, we want to present our structured approach of managing the scale-up of leads to GLP tox and candidates to IND and first-in-human trials. In addition, we hope to listen and learn from you and add to the experience of all the participants today. My name is Marcel Veltrop, and I'm the president of Jubilant Biosys. I've worked in the global pharma industry for over 20 years. The majority of this time was spent on providing scale-up services for NCEs to innovators in various phases of development, from preclinical all the way to phase 3, as well as commercial launch. Biosys is both a fully integrated discovery CRO as well as an early phase CDMO. These are very different but complementary skill sets. The discovery engine delivers leads and candidates which finally need sufficient quantities for GLP tox and then GMP IND studies. In this webinar, we will focus specifically on the important transition from discovery to IND preparation and how to diagnose the right strategy for the unique routes coming out of medicinal chemistry. The key questions we will focus on are: when does process R&D and scale-up play a role? And how do we prepare for the GMP phase 1 supply? In the next section, my colleagues Vikas, Global Head of our Noida Operations, and Purshotam, Director Process R&D in Noida, will take you through our approach in more detail and share some case studies which have demonstrated the benefits of our approach. We will then proceed to the discussion and Q&A and hope to have many examples and questions from you from which we can all learn. With this, I pass on the presentation to Vikas and Purshotam.
Okay, thank you Marcel for that very pleasant and lucid and sharp introduction about the whole life cycle of the pharmaceutical product, right from the beginning to the end of the manufacturing. I will take you through the webinar structure, which will include some of the introduction to the Jubilant Life Sciences and then to the Jubilant Biosys. That will be followed by the IND case, the life cycle, shorter life cycle of the IND case, and then that will be followed by the first 100 grams. Now that's the area of our focus today. We will talk about multiple aspects of the 100 grams case, which reflect into the later on multiple aspects like the scope definition, the scientific psychology, the cost of the product, and the regulatory aspects and whatnot. But bringing back to the end of it, what is our 4D philosophy and how we handle this matter very differently at Jubilant. So Jubilant Life Sciences, if I have to introduce, it's a publicly listed organization. It's $1.3 billion of revenue according to FY19 numbers, and it is a publicly listed organization. Coming to the Jubilant Drug Discovery Services, which deals mainly with the early stage discovery, that is the concept level and the preclinical discovery stage, which we started with the target validation and then the medicinal chemistry, including hit identification, lead optimization, and we go all the way up to the candidate selection. We don't stop there. We also go forward to the developmental stage, which is the focus of today's discussion. We have over 750 scientific team working at both the sides, supporting the whole preclinical discovery and the development. We were established in year 2000, and the strength that we have is that since last 20 years, we have been delivering the integrated success stories. We have delivered over 70 integrated projects and more than the milestones. I would say over 80% was the success rate of our achieving the milestones, meaning to say we were successful 80% of the times delivering the needs of the drug discovery industry in the early stage and now also the later stage developmental work. Okay, so let me bring you to the most important part of this discussion, and we want to generate the queries and throw some questions for the audience so that we can learn more from your questions and generate some kind of discussion. So if you look at a typical life cycle of an IND, the transition from medicinal chemistry to the scaling up I&D group is the most critical part of it. Whereas most of the aspects which come in later on while addressing the I&D are very well discussed. This phase of transitioning, and it's a small tech transfer from medicinal chemist to the process chemist, is the most critical one when it comes to the synthesis of first 50 grams or first 100 grams of the compound. That is the area in which we believe we can manage most of the questions that are going to be raised later on. The questions related to the cost, the questions related to the quality, the timelines, and how we can save on multiple aspects if we just acted in time for all of these things when we do the first scale up of any of the product. Whole story actually revolves around the cost, scope, and the quality. In the early stage, I have seen there are cases where the cost is not much of a concern, and then quality is focused upon, neglecting some other aspects. This is the discussion therefore which will generate some queries around it. Cost, quality, and the scope is like a love triangle. You see, two of them are always comfortable. When the third aspect is to be investigated, that creates little trouble in the situation and then puts a burden of decision making on the whole team. We at Jubilant look at even the fourth aspect, and we say that's the dimension of time. That's our fourth dimension. Looking at all the aspects at the right time is the major aspect that we believe can actually lead to the success of the I&D. And this is what I'm going to take you through. I mean, there are other aspects. There are aspects like the control of variables, the safety of the process, what are the purification technologies that we are using for that matter, or are you taking crystallization into account? Have we considered polymorphism right in the beginning? And should we or should we not? Whether the process is feasible? What is the economy of the chemistry and of the process? What is the atom economy? And most of these aspects, instead of getting discussed later in the life cycle, it is always advantageous to do it beforehand when you are doing the first few scaling up. And that is what this slide actually shows you. If you look at all of the aspects, they are mainly chemistry, regulatory, and the process related aspects. Now, these are very trivial aspects. Chemistry issues are, some of them are more related to the route of synthesis, whether there are multiple steps in the synthesis. Do you have a racemic product? Are you using the chirally selective route for synthesis, or are you using purifying the chiral enantiomers at the end of it or in between? Are there potential side reactions which could be disastrous later on? What is the purity that you are looking at when you are getting this first 100 grams? Are you using the unsafe solvents? Are you using some of the reagents which could be later on harmful to the process? So all of these chemistry aspects also spill over therefore to the regulatory aspects. For example, if there is a process which is generating the impurities and that is neglected right on, the material which is used for generating the biology data is going to be not appropriate. The biology data on the later stages may stay invalid just because it is producing some of the impurities which are, say for example, equipotent or sometimes more potent off target, generating more of the toxicities, and that generates multiple queries. The residual solvents, the solvents that you are using, is the process able to take care of them later on? And these are the regulatory aspects that we need to look into. Are we having analytical methodologies? Can we develop it going forward? So it is more of foreseeing the problems that do not exist right now, but they are going to come. 80% of the scale-ups that go into 100 to 500 gram scale mostly hit the next stage, and the decisions are taken positively for the I&D. And if we neglect it right now, what happens is these things grow up later on, giving a lot of disadvantages on the cost and the quality, mostly also on the time front, because the data needs to be reproduced. The process aspect for example, are also very important. Have we considered the engineering aspects? What are the techniques which are being used? Are we using some very critical reactions, very tough reactions, very intricate reagents which could be difficult to be handled at a larger scale? Are there reagents which are potentially hazardous to handle? Have we done the process safety in time? And are we using the right technologies which can be scaled up in the later stages? And these are the questions which often arise. So to say, if you look at it as from a different perspective, you can also split the same problems from the perspective of a route and the process and the aspects of the process, and then that becomes a very good conglomerate of queries which needs to be addressed later on. Why not right in the beginning? For example, route is often a debatable aspect because it's a war between the medicinal chemist who's leaving the project and the process chemist who's sticking on it. So whether the route is right, whether the reagents used are correct, and what are the differences of opinion between the medicinal chemist and the process chemist. Have we balanced it before we go on to the next step and take a decision? Development of the process, again, is the same dilemma. I do want to develop it, for sure. How much? What is the right development? Do I just do a fit for purpose and leave it for the next stages to be developed? What are the aspects that I need to see today and there is no option? What are the aspects I can leave it for a couple of months down the line on the decision? And then there are other process aspects like the number of steps, the safety issues, and all other aspects. What we do at Jubilant is slightly different, which is not in tune all the time with the rest of the world. I mean, we have the passionate chemists who keep a track on every medicinal chemistry project. You can tell what is going forward. Right in the earlier stages, when you're scaling up even to the 10 and the 50 gram scales, you can see with a constant communication from the collaborators and from the medicinal chemists and from the biologists that there are some compounds which have higher possibility of going forward. They are the winners in the race. So we start looking into the process aspects right then and there. We look at the simpler aspects. For example, what are the isolation techniques we are using? Are they going to be good and scalable? Because these things also matter at the 10 gram and 50 gram scale. When it comes to 100 gram scales, therefore, Jubilant is much well prepared in terms of the process, the route, although the route may not be the one which are going to be used at a multi-kg or say ton scale, but it is still very close to the reality of a dream route of a process chemist, and that's the difference that we bring out. All of this thought process and the complex thinking and the attention to detail right from the beginning and at the early stage gets you to the stage of a right mixture of philosophies at scaling up at the first 100 grams and beyond, thereby making the R&D more successful. Okay, so let me take you to the details of some of the aspects. Like there is a cost, there is quality, there are regulatory aspects, and we will just see one or two questions all around them. So when it comes to the cost and the quality, the early stage arguments are very different. The typical one that comes with, I'm fine with the higher cost. We just have to make 100 grams, and that's okay. We can use the current process, for example. But then, if we neglect the cost and the quality aspects at this early stage and take up the faster route for synthesis, apparently faster route for synthesis of 100 grams, we are going to land up in troubles later on. It could be regulatory aspects or, when it comes to CMC anyway, we have to do some detailed experimentation. Minimum experimentation is required. Why not do it beforehand? For example, when we look at the quality of the material that is needed for different purposes. So when you're making 100 and 500 grams for say non-GLP tox or the efficacy studies on non-GLP, do we really need the high quality material? But are we considering also the root aspects? And should I consider it? Should I not consider it? When it comes to this discussion, typical argument is, why would I invest in the process development or the root scouting activities right now? It is not required. I simply need a material. But then we certainly in this philosophy tend to lose a focus on the route of synthesis, the potential impurities that may be formed, and then whole lot of other aspects of the process that is going to be employed going forward. The question is like, can you look at an ideal timeline for filing the IND? No, that is not possible. However, with a given situation, at every stage, defining the right scope is very important, which generally tends to impact the cost and the quality. And the time is constantly running in the background. That's not something which can be compromised. Neither can be quality. And therefore, these dilemmas needs to be addressed. For example, if you look at the regulatory aspects, it's a very dynamic situation with respect to all the aspects. Look at the dynamics of the quality requirement from the regulatory perspective. The quality requirement for non-GLP tox, anyway there is no regulatory control, but there is a strategic control that is needed there. I have seen, for example, the cases where too much pure product which was used for non-GLP tox became a hurdle because we could not produce that pure material after the GMP development and for the IND submission. There were a few impurities which we could never get rid of when we used crystallization for that matter. Neither is it a regulatory requirement. Are we burdening ourselves with the extra quality requirements at the early stage, which actually puts us into the compromised situation with the quality later on, or vice versa? We cannot use very impure material which has impurities which are not going to be there in the later phase. Even that can put us into the troubles with respect to the biological data. Therefore, it's very important to tie up the purpose with the quality requirement, and that's what is the strength that we have. Along with our collaborators, we constantly debate and question and generate the queries to solve these problems early on. Again, is there a ideal time for IND filing? There isn't, because it is dependent on multiple activities. There is a biology dependence, there is a toxicology dependence, there are non-GLP studies which are being taken care, and then there are funding situations, especially when it comes to the biotech industry, which is very vibrant and speed hungry, and therefore taking decisions at the right time matters a lot. What is the difference that we make it to this whole process? When we look at the history of Jubilant Biosys, we have been serving drug discovery industry, the preclinical as well as the developmental industry for the last 20 years. 60 integrated projects means a lot. That means the whole team is completely aware of the early stage and the late stage problems and the issues which we have a knack of identifying, at least, if not solving at an early stage, which is very important. A very experienced team therefore looks at your chemistry early on and then starts tackling it. For example, if we are working on some integrated project right from the discovery and it transitions it to development, our medicinal chemists actually keep in tune with the process chemists even when they scale it up to 10 grams and 50 grams of intermediates, for example, and then they discuss and find out the solutions from perspective of a scaling up. How are we going to purify, for example, this particular intermediate, or there is a reagent which is going to be troublesome simply going at the 1 kilogram scale, and these are the aspects we are able to therefore tackle before even if it goes to the 100 gram scale. And when we are at 100 gram scale, we take care of most of the aspects which are going to come in future. when it goes to 1 kg, when there is a regulatory control now on the process, and all of these aspects of the process are very well therefore taken care. So the strategic planning of the dependent and the independent activities in the process gives you an edge when it comes to going forward with the regulatory submission. And then this whole debate, actually what exists at the first 100 gram scale is really very interesting. There are two views here. There is a view of a medicinal chemist who has correct arguments from his perspective, and then there are arguments from a scale-up chemist's perspective which are also correct from his perspective. For example, there will be an argument, oh, I've done immense work on this route of synthesis. Why would I just give it up? Let us make 100 gram with this route. Sometimes there is simply emotional connect here, which is very difficult to break down. We say, oh, we have a complete control of supply chain. Why would I change the route and look for some new materials which I am going to use, and then I have to go all about the supply chain problems? Some say we have researched very well and we are using the best of the reagents for the synthesis, but are these reagents scalable? Are these reagents good when it comes to, for example, scaling up to the larger scale? And for example, chromatography. There are multiple steps where there is a column chromatography, or where you want to use the SFC for purification. And then the general thought process is, because the technology has scaled it up, we can even purify kilograms using SFC. But is that a correct approach? And what we will miss if we take that approach? And then there is therefore a counter argument which often comes from the process chemists. They say, oh, no, I'm not going to use this reagent. Look at the solvents you are using, for example. ICH does not allow this. Oh, you are using metals in N-1, N-2 steps. How am I going to take care of them in the final API? That's going to be the hurdle going forward. So these are the aspects which the process chemist fights to convey to a medicinal chemist. And then the argument actually is so productive, it brings out the best and leads to the right decisions, which are maybe developing the route for a particular segment, or developing all of the route, scrapping the everything on the science that was done in the medicinal chemistry phase and taking up on new stuff, looking deeply into the purification technologies and at the cost and the supply chain aspects of it. That is something which is very important. Let me give you two small examples and how interesting it is to take these decisions early on. This is a very interesting case. It was a IND which was high pressure. We wanted to file it in 11 months and we did it actually. This is an oxapane ring system which is fused with 2 aromatic heterocycles. And the synthesis philosophy was closing the oxapane ring. The moment you close the oxapane ring, we generate 2 chiral centers, and that's the trick. And there was a lot to be done here, in fact. So the medicinal chemistry route you see on the top employed actually the racemic alcohol, which was closed using a Michael reaction. It was an internal Michael oxa-Michael addition reaction, which produced the final desired diastereomer, that was SR, which was purified by SFC. However, look at this reaction. This reaction uses DBU and potassium carbonate and employs SFC for purification. The product formation is only up to the tune of 35%, the desired isomer, desired diastereomer. And then there are 3 unwanted products. So there is a syn product, which is also a major, it's a 30% loss to the process. And then there are 2 side products formed. One of them is a retro-Michael product, which could be avoided. And then there is some product which hydrolyzes, gives an acid. Again, that's a strict no. Because the last step after this is only giving the API. We decided that we want to work on this project and we are not going to use the SFC for purification. After a debate of a day, we agreed on working on it. We screened multiple reagents and look at the beauty of the chemistry. We stumbled upon something so simple. Sodium ethoxide and ethanol at room temperature actually gave us something which was not expected. Of course, there is another change here which was done on the earlier part of the process. We used a chiral alcohol. Used the simpler reaction conditions just to produce 90% abundance of the anti-product which was formed in majority. And then there was only 10% of the syn product which was formed. We completely got rid of the retromichael product and there was no hydrolyzed product produced in this step, thereby improving the efficiency of this process 3 times. And given the fact that this is a n-1 step, it was actually a huge cost saving for us. This is the beautiful example I often quote from our recent chemistry. Look at the other one. This same product would then be hydrolyzed to an acid completely, and then we would do amide coupling reaction. It was a straightforward H2 and the respective amine in dimethyl formamide, which also needs water workup, by the way. But then there were 2 steps, and the process chemist doesn't accept this. They look for reducing all of this. And it was a very interesting suggestion which came from one of our process chemists. He said, okay, I am not subjecting 2 reactions. There is a possibility of racemization. I am not employing longer durations and too much of shocks to my final product. Well accepted. The suggestion was doing it 1-step conversion of ester to amide. We simply used trimethyl aluminum and got a beautiful conversion from ester to amide. It was a very clean conversion. It gave us a ease of isolation, and the isolated crystalline product, which also was subjected to crystallization for a morph though, but this step was reduced. So we reduced 2 steps into 1. If we gave up early on these 2 steps and took up with a fast pace, took the help of SFC, we would have not reached here. So these are the 2 examples that I quote from our recent examples. So this is the difference that we make with our 4D principle and the philosophy to the whole early stage process development, which takes us through to the IND submission. All our facilities, the manpower and the experience which stands behind the delivery is something that we are really very proud of. But we are also proud of our infrastructure that supports all the processes and confidentiality aspects. We have very high safety standards internally, therefore we look at the safety aspects of the process very early on, and the compliances are met very early on. No compromises there beyond the quality. These are the major aspects that we look at. We have fully access controlled facility, including the scale-up facility, which takes care of the confidentiality of the matter, not only in the early stage but also during the process development. We have the operations which are designed for high speed of delivery, saving the time. We have the integrated operations which considers the quality control teams, the project management teams, the supply chain teams from early on, and they are the integral part of the scientific delivery. This whole teamwork actually makes us unique, and our 4D philosophy of looking into each aspect early on, much before it strikes, it's very important. I cannot give a better example in the pandemic time of COVID-19. Remdesivir is one of the bright stars on the horizon. It has been developed now, it has been accepted for the treatment, and there are multiple organizations across the globe trying to catch up with synthesis of this beautiful molecule which helps us win over the COVID. But when you look at the process, some of the aspects I believe could have been solved early on. There are complex Grignard chemistries which are employed in the early stages of the synthesis, which is becoming a stumbling block. When we realized it right into the pandemic, not having solved this matter early on, we are now delaying the reaching of this product to the patient when it is needed the most. The patient is really actually waiting on the bed for this product, and the delay in this developmental stages is actually piling up the cost, increasing the time to the patient, and delaying and in fact denying the solution which is already existent. So with that, I would conclude my presentation. Thank you so much for your patient hearing, and we are getting into now the discussions and the question and answer sessions which will be guided by Marcel. Thank you.
The number of questions which arise when we deal with the judgment of moving from the MedChem phase to the scale-up phase. I want to invite all the attendants to contribute either an example or a question. So you have a Q&A button on the bottom of your screen. You can type in, we will see that here, and we will host the questions between Vikas, Purushottam, and myself. So anybody has any questions or comments or suggestions here? I see Anil is asking a question about the importance of experience in your team, Vikas. And do you agree that getting intervention by the right people at the right time is critical in scale-up efficiency?
Yes, indeed. In fact, these kind of interventions are needed. We do plan it. In fact, the views of the medicinal chemist and also the process chemist at the right time are the ones which actually make us think, make us debate, help us come to the right conclusions, and guide us in the right path, ultimately.
Okay, so clearly experience is very important. And if I add to that from my own experience in this industry, it is learning. This is not something that I think textbook can give you guidance on. You need to experience a lot of unexpected events, scale-up problems that arise, and that builds a body of expertise that you can apply to the next project. So I think to answer the question fully from Anil, it is vital to have people that have gone through the motion, that have a number of scars and bruises, as I call it, on the skin. And that is how robustness is built in future. I have a question here from Rodolphe. Rodolphe is asking because broad ratio between FTE and FFS services. Obviously, scale-up is deemed an FFS service. So what is your answer there?
For a typical IND, I would say, Rodolphe and Marcel, that it would be about 30% of it will go in an FTE mode, because that's what we do for our route scouting as well as the process development phase of the project. However, the rest of it, which is either non-GMP manufacture or GMP manufacture, typically goes into the FFS phase. So I would say 70-30. Yeah. If that answers the question.
Okay, I think we see a second question from Rodolphe. He's asking, there's over 750 employees. With regard to PR&D scale-up activities, how many people are working on it? So Rodolphe, if you add, so PR&D alone is not completely handling scale-up. It's a broad team effort, and you're easily looking at 60, 70 people that need to be involved, from supply chain project management, analytical engineering, process R&D. Of course, as one of the primary activities, we always include our MedChemist for knowledge transfer into those teams as well. So to answer your question, it's about 10% of the workforce right now is fully active in scaling up for GLP tox and IND phase. Let me see. I see a question here from Sumama Ghosh.
So what are the purification processes you prefer for scale-up? Vikas or Portugal?
Yeah, for the purification technologies that we use during the scale-up, we depend nothing else than crystallization. That would be the ultimate and the best option for us. We have always stumbled upon other options. In the non-GMP phase also, we have seen sometimes that we have to take the help of column chromatography. Doesn't work when we go ahead. And we have seen in 10 odd GMP INDs that we have filed, if we attempt, we eventually succeed in getting to the crystallization. That is the best and the most preferred way for purification in the scale-up, non-GMP or GMP.
Absolutely, and of course, as you mentioned, any type of purification, prep-based PLC, SFC, column purification, those are typically the brute force methods used in a MedChem process. So when I look at the average MedChem process, you could be 10, 15 stages of chemistry. You often find several steps that require some form of column purification, chromatography. But ultimately, you are developing a crystallization method for robustness, and that is the important later on. Here I've got a question from Alexandra Lopez. She's asking how crucial is the use of design of experiment and kinetic modeling in scale-up processes. So this is an important question, and this is actually guided by ICE 211 guidelines. So clearly in phase 1, we are not doing DOE or kinetic modeling yet. I think that is deemed far too early. But phase 2B onwards, design of experiment and modeling is important because that goes into your phase 3 validation batches. So the amount of process R&D increases over the various phases of scaling up. What we are discussing here is typically the tendency is to not do enough from discovery to first scale-ups. And what we are arguing and promoting is that you should do a very tailored assessment and do the right amount of development so that you benefit from it in terms of speed, cost, and robustness when you have your IND package ready for GMP phase 1. So at this stage, we are not yet doing DOE or modeling. That is still considered too early. I hope that answered the question. I have a question from Manas. How early do you think, as per experience, we start working on optimizing the route or work on PR&D? So, Manas, the question here, or the answer here is really explicit or implicit in the presentation that Vikas gave. Every time we look at a MedChem process that is an individual assessment and evaluation, we look at what are the challenges, and if there is something that we believe can scale up without much hassle, we don't want to spend time. So it's a judgment, and I've had this friction with a number of my customers in the past where I was insisted upon by the customer, no, you don't develop anything, just take the MedChem route, and we do a 9-month pivotal doc study. Which was an absolute disaster. It delayed by 3-4 months. It was a 12-step process. And at the end, we found an impurity that went back to step number 1. So there is a lot of evidence, empirical evidence, that suggests that you need to start and not take anything for granted. So I would say the moment that you enter into larger vessels, that by virtue of your process and overall yield, you need vessels of 50, 100 liter. That is when process optimization is mandatory because you will run into scale-up surprises almost 99% of the time. That is my experience over the past 15 years active in this business. I see Anil. How much of your work is innovative versus biotech? I assume means big pharma versus biotech. And how much complexity is typically involved in the scale-up projects we deal with? So what I'll do is I'll take the first part of that question, and Vikas, you can take the second part. So the majority of our activity is actually for the biotech sector. And why? We see the importance the biotech sector has for speed. is the highest in the industry. We work with companies in Japan, Asia Pacific, Europe, and the US, which are the innovation hubs, of course. And it is notably the biotech who are under resource constraints. They have the need to run to a milestone fast. Typically, that is the pressure is not enough development is done. Because the complexity of scale-up that we see, what would be your impression based on a whole host of programs that you've been involved with? Is it low complexity, high complexity?
Yeah, thank you, Marcel. I mean, you said it. Of course, in the earlier phase, there is a 100% contribution in terms of innovation, because these are the NCEs which were never ever synthesized, and those are made in the lab. Now, during the scale-up, what is the component of innovation and what is the complexity? When I see nowadays, the complexity of the final compounds is increasing. The multi-chiral compounds, the ring-fused compounds, and it's pretty complex. When it comes to medicinal chemistry scale, the things are very easy because you can afford to use any kind of reagents. You have no issue about the safety. You have no issue about the costs. And that's where the innovation component comes in while scaling up, to think very differently, to take the risk, to try the best of the possible routes and get to the one which is going to give you the best quality, which is going to be the best of the cost, and it's going to be the safest one. So I would say there is a huge level of complexity there, not only in terms of synthesis, but also in terms of the purification aspects. The chirality adds a lot of complexity. Look at the remdesivir example, Marcel, that I gave at the end of my presentation. This is the newest compound on the block, and look at the chemistry that it employs. The internal double Michael reactions in some of the stages, and sugar chemistry, and it's a huge complexity of chemistry that ultimately produces this nucleoside analog. So there is a huge level of complexity in the newer chemistry, I would say.
In addition to that, Vikas, thanks for that. If I look back at the literally hundreds of routes I've looked at over the years, you see a steady increase in complexity. Even if you look at the FDA approvals for small molecules in 2019 and 2018, then barring a handful of what are relatively simple molecular structures, you see complex molecular structures with chiral centers, with fluor compounds, on average, easily 10, 15 steps of chemistry. So that precision is also linked to the expansion of therapeutic areas in niche areas, orphan drug status. And you see a higher candidate success rate when molecules are actually more precise and well designed. And that simply means a diverse molecular architecture, and inevitably that puts pressure on the process R&D chemist, who has to take that complex molecular architecture into a process that can be made ready for GMP in human dosing and beyond, of course. And the moment that you do sufficient amount of work, you benefit from it. And again, to your point of remdesivir, it would have accelerated the availability, and it would have also made it more affordable for global populations, because right now it's still a pretty expensive molecule. I have a question from Ravi here. Does the 4D approach bring not only speed, but innovation and effective project management that controls cost as well as quality? So, because you want to have a go at this?
Of course, I would say the project management forms the backbone of the whole life cycle, right from the stage when we start planning about the route scouting and till the delivery of a GMP material. Project management contributes in the material control, the cost control through the material, the resources planning, use of the right facilities, and checking of the safety aspects of the whole process, whether they are being done. In a way, our project management team in this domain of business actually is more than a skin dip. In fact, they help us be on track with respect to the timelines, the cost, and the quality?
Yeah, I think you cannot achieve speed with all the criteria that have been presented today unless it is packaged in a very effective project management approach, which manages the scope and input of all the critical departments that are on the critical path of the project. And in terms of innovation, of course, innovation has a time penalty attached to it. So it needs to be the innovation that innovates speed. But clearly, without bringing together the teamwork, you're not going to get the amount of speed, because it'll be a sequential event of troubleshooting from one department to another. That has been my experience over the years, and only true teamwork can manage speed with all the right criteria attached to it. I have a further question here. I think it has been addressed. QBD and DOE is an element of QBD. So that is also an aspect which is typically not seen early on. But QBD as such is a useful concept, and there's nothing wrong with applying the thinking of QBD, which talks about what are your critical quality attributes, what are your critical process parameters. Because the whole point of process R&D is that you are building up the body of knowledge to address unknown process sensitivities. A process can trip off, a reagent can cause a yield to be low, or you have a reagent quality which gives rise to impurity formation. So these are all aspects that QBD teaches you. Of course, QBD goes further. It goes into the statistical software. I think that is not critical in this stage yet. That is more for phase 2B and 3. It's mandatory in those stages. But the concept of QBD, I think that makes sense to apply early on, and that drives your thinking in an efficient fashion. I have one question here from Nigel. When do you evaluate genotoxic potential impurities of the process, and how do we do that, Vikas and Purushottam?
Purushottam, you would like to take it?
Yeah, we take it up actually in the beginning of the root scouting itself. After root scouting, we take it the vitogenic impurity assessment. All the reagents, solvents, and intermediates will be taken up into the consideration of this assessment. We'll do the ICH M7 assessment by following the Q3A controls, and we do the computational risk assessment by statistical softwares like Direct, Nexus, and LeadScope softwares. Apart from that, we follow the ICH M7, the control strategy of option 4, where we study the purge factor. Also, we take into consideration for the classification of the impurities of 1 to 5.
Thank you, Purushottam.
Okay. Thank you. And the second question that Nigel has is, do we have the necessary infrastructure to carry out the process safety studies?
Yes.
Yes, Marcel. We first check the Derrick, Breathric, and MSDS, followed by the reaction safety studies, initially by DSC, followed by the RC1 studies. We have all the facilities in-house.
Okay, very good. Let me turn this back to the audience. Is there anyone who has an experience that deviates or wants a challenge? I have a question here from Ross. So how has COVID-19 affected your ability to deliver services? We need an SOP, but also some flexibility without losing quality. Have you managed well during this time? So I think we have been busy with COVID impact since it started. It has, in the initial phase, impacted stability because simply people were not able to reach office. Right now, we have implemented a whole set of stringent, what we call good COVID practices. And that has allowed us since about 6 to 8 weeks to run at near 100% capacity. I'm talking 98, 99%. On one day it can vary with a few people here and there, but None of our projects since being at this level has suffered, and we have things under very good control. It's work from home, it is distancing, it is shift systems, mask wearing, sanitization with the high frequency. So this is both mandated by the government as well as by our own corporate safety team. So there is a daily team that tracks everything around the COVID to make sure that we can operate as a essential industry. So Nigel and Ross, I hope that answers the question. Back to the audience, are there any other contributions, experiences someone wants to pose? It doesn't have to be a question, it can be a statement or a challenge. We have several continents on the line. I see people from quite a few from the US, Europe, as well as India. So this is a global event. I think then we are, if I don't see any further questions from the audience. Then let me pass this to the end of this webinar. I want to thank everybody who attended for participations and patience, as well as the host of questions that were raised. We were delighted to have you on the call today. We hope you have been giving you a little bit of more insight into how Jubilant Biosys approaches this in a very much tailor-made approach around every single project and the experience we have around it to help drive scale up in the fastest possible way. I've been part of such events in the recent weeks, of course, and one important feedback that I got from especially big pharma in-licensors is their biggest complaint when they are looking at licensing from particularly the biotech sector is that often the CMC packages are too thin and that fixing them would take another 9 to 12 months. And that time loss actually erodes the deal value of the biotech. So this is another important area. The value of your discovery and development improves if some base attention has been given to the supply chain, that it is robust and it can stand the test of time to go to phase 2 quickly without a major rework and without running into impurity roadblocks or otherwise. So that's with the final words I would want to leave everybody. If you have any specific confidential questions, you are most welcome to reach out to our global BD team. You can find us on the Jubilant Biosys website. And we hope you enjoyed this. We hope that it was insightful for everybody. Vikas, any final comments from your side?
Yes, indeed. Thank you, Marcel. You said it. And this is one very critical area which actually impacts not only non-GLP, but also the GLP scale-ups in the later stages. And we wanted to generate some queries. I hope we are successful in generating the queries, and even after the webinar, we would welcome all the questions to our emails. And yes, thank you to all the attendees for the time today.
Okay, very good. Have a good day, everybody. We're signing off here. Bye-bye.